Hi everyone. Hi, hello and welcome again. Microbehunter here. Just checking a little bit if everything works. Okay, I think I can see myself online. I hope that you're also able to hear me. Yeah, so yes, I can hear myself, which is nice. And yep, I think everything seems to be set up, and I want to wish you a nice Saturday again. Thank you that you are joining me now again for this Saturday Microscopy live stream. I see that a lot of people started to join in again. Yeah, at the moment, let me double check. We're already 36 people more are joining in. Pretty nice. And yeah, usually during the first couple of minutes, I just say hello all around the world to give viewers a little bit a chance of starting this video here. So yeah, so hello to North Carolina, the United States, Hungary, Germany, Belgium, the UK, France, Netherlands, Norway, Germany, England, Texas, Finland. Okay, really nice. So UK, Ireland, UK again. Okay, Lebanon. So hi everyone again. Officially, yeah, from hello from Central Europe. Look what I brought along here. Yeah, beautiful. I love these things here, right? No, not very nice smelling, not very appetizing. An orange here, a moldy tomato, and a moldy strawberry. Not the first time that I'm putting mold under the microscope, but I was successful in actually also growing the mold here on a Petri dish. So I'm going to show you how I've done that in case you want to have a controlled culture. I also brought along again a couple of my stains here. So when we put it under the microscope, we're also going to stain it a little bit today. Yeah, and we're going to basically see where the live stream is going to take us, and maybe you have some requests or so, and maybe I'm able to yeah to do those as well. In case the unfortunate thing happens that the live stream becomes interrupted, this has happened rarely, but it did happen before. Then I will try to restart the live stream. It could be that it will be in a new link, so you have to be patient maybe for a couple of minutes, and then I'm going to try to go online again. Okay, so this is simply a a thing I just want to tell you. If you are new to the live stream, it's like this. There is the possibility to include a question. You can basically type in questions into the chat. It will take a couple of minutes for it to appear. And if you want me to, yeah, then I'll try to respond to it. But usually after 1 hour, I will try to answer all of those off-topic questions. But please, if you want to send a comment to me, then please put down at Oliver or at Microbehunter so that it's easier for me to find the questions. So we're going to start it right at the beginning. Yeah, so just every now and then I'm basically stopping a little bit to read the comments here. Okay, yeah, so hello from Spain again, from France. Okay, what are some safety concerns here? Very important. Yeah, safety is important, especially when dealing with microorganisms and also with spores. And I did a little bit of research here because this one over here, the fungus that attacks those citrus fruits, lemons especially, and also oranges, called Penicillium digitatum. I hope I pronounced it correctly. Yeah, it's basically the same genus of fungus that was used to actually extract penicillin. So penicillin is called penicillin because the fungus is called Penicillium. And if you do a little bit of research on that, then you're going to say, yeah, of course, it's not always healthy to inhale those spores, especially if people are allergic. But there are also not any known cases where it's actually dangerous. You should not be eating these fruits, right? But these fungi will, of course, harm fruits, but not generally not humans, okay? The reason why you should not eat moldy fruit is because not necessarily only because of the mold itself, as disgusting as that might be, but because the mold, of course, produces toxins, so-called mycotoxins, and they can be quite harmful, and they're given off into the fruit. So yeah, that's the thing. So generally, how did this actually work? Okay, so what I'm going to do first is I'm going to, why not? I'm just going to show you how I made this thing here, okay, this Petri dish. And as a matter of fact, it's about 2 days old. Difficult to see because the contrast is so low, difficult to see under the video. But there is quite a bit of fungus already growing on here because I transferred some of the spores from the orange to the Petri dish. We're going to have a look at this first under the stereo microscope. Again, not going to be very clear because simply because it's the contrast is not high. Yeah, but basically this is how it looks like. And yeah, you see that. Maybe I'm gonna turn on the light from the side. Just a second. Maybe this is going to do a little bit. Increase a little bit I don't know, maybe because when the light comes from the side it casts a shadow. You can see that it does look a little bit orange. That is not slime mold. for those of you who've been noticed a little bit, that's a little bit some of the orange juice that you're seeing here because I used orange juice as a growth medium. Yeah, so here you see it quite nicely. So I'm going to be transferring this then also to be on a microscope slide so that you can actually see that. Because, and that's also one of the things that I would like to show you, because we can actually watch the cells grow. Okay, yeah, that's actually quite time-lapse. Yeah, you can actually watch them grow. Here's quite a bit. Yeah, so I'm just going to show you first how I've done that. It's pretty easy. Yeah, but as always, you always want to be a little bit careful not spreading any spores around. Okay, every now and then I'm going to read because it's really safety concerns, right? You don't want to smell those, but I'll be also honest with you that indeed, if you have an apartment maybe where there is moisture and where there's fungus growing in the wall, that's not healthy either, right? So yeah. It shouldn't be an issue for people who basically have a good immune system, but generally there are some people who are allergic. So I'm going to show you how this actually works. And what I have done is, let's move this to the side here. So the process is a little bit similar to the slime mold thing. So what I've got here is I've got a Petri dish. I'm just using the glass ones. You can also get disposable plastic ones. Look what another thing that I've got here. These are called coffee filters. And I bought the white ones, but maybe I should have bought the brown ones because simply for better contrast. Okay, so yeah, I'm just, you see, you put basically the coffee in here. And what I'm doing is I'm just going to leave them together like this, and I'm just going to use 2 of them. And then you simply turn it around, and I'm just going to, I need something to draw. Okay. I have a dead spider. Ha! Do I have any tips on if you want to observe spiders, you want to use a compound microscope because they look quite nice. Not a compound, my bad, a stereo microscope. Because otherwise they're too big, and you have to, or you have to pick it apart, and then you can look at it. Or you can use a compound microscope and use the low power objective. Yeah, to observe the spider from top by using a flashlight. So you get the idea. What I have to do now is I have to cut it quite a bit smaller, okay? Because actually quite a bit, maybe about 3 millimeters smaller. And you see that I'm basically cutting both at the same time. Okay, and I can be quite generous about this because I do want that the filter, is flat. In the Petri dish. I think maybe there's a corner here. So that's step number 1. Yeah, of course there are 2 of them here. I'm just going to leave them together. Goes in here. See? And we're almost finished. Okay. So let me throw this away. The next thing is, look what I got here. Fresh orange juice. Freshly pressed. I did put it through a sieve to remove the pulp a little bit. You don't have to do that, and there's still a little bit of pulp left, right? But that's going to be our growth medium, right? And what I'm going to do then, either I could pour it in, obviously, but that is a mess because I'm going to spill quite a lot. So what I'm going to therefore do is I'm going to use my pipette, and I'm going to simply use a pipette, and I'm going to add in here. I'm just going to do something like this, and I just want to make sure that I soak it completely, but I do not want to have any standing water, any standing liquid. So usually what I do then is I kind of try to press it flat a little bit to remove all of the air bubbles. Simply for convenience as well. And that's pretty much it. And yeah, air bubble's gone. It's not so important. I usually tip it over to the edge and I remove any excess liquid. Yeah? And then if it starts to evaporate, it will start to evaporate, then you can always add a couple of drops of regular water to the side, and it will suck into the filter paper, and then it'll be fine. Okay, so I'm just going to throw the liquid away here again. So, and we're actually almost ready to go. The next thing that you need is, and I like to use toothpicks because I can throw them away later on. What you do is you now go to the orange, and what you do is you carefully simply from the gray part, because this is where you have the spores, simply do something like this. And scratch off a couple of the spores. You know what I'm going to do now is I'm also going to show you this tip here under the microscope, okay, under the stereo microscope, because you might actually see under the stereo microscope that, here it is. Where is it? It's too bright. It's a little bit of a pity. It's a little too bright. Let me see. Let me see this. Let me turn off the other one. Okay. You see all of those gray things here on the tip. These are the spores. Yeah, and they're scattered over the air. And when they settle down, then you're gonna have a new fungus. What I'm doing right now is these spores. Oh my gosh! Thank you very much for the donation. Thank you very much. Okay, so what I do now is I do the following. Look, I just carefully streak it over here. I mean, of course, some people like to do smiley faces and I don't know what, right? And you're simply transferring the fungal spores, and yeah, you pick up some more of this. Now, why would anyone do that? What's the whole point? Yeah, good question. Well, the thing is because you kind of, I'm going to show you in a second, because we can get actually quite nice fungi where we can actually observe the growth of the cells. And I made a time-lapse video, which I also would like to share with you. Okay, and this kind of also demonstrates indeed that that the spores are quite abundant. And one of the things that I also want to do is, and I was not quite successful yet, is I want to actually put the spores directly under the microscope. That's easy to do, right? But also see them germinate under the microscope. But the thing is that they need quite a bit of oxygen. And when you put the spores on a microscope slide with a cover glass on top, then sometimes there might not be enough oxygen. So that's a little bit the reason. So, and that's basically all I've done. This here goes into the trash. Lid goes on top. You wait 1 or 2 days, and then you basically have it growing on here, right? And that's pretty much it, right? And of course, now you want to know, how does this look like under the microscope? And we're going to do that. And no, I'm going to show you first the video, and then, I'm going to simply give it away because the video is a time lapse. And then this is how the video looks like. I made it, I think, 50 times faster or so. And if you look very carefully, you're actually going to see that inside of these cells here, there is movement going on. So there is quite a bit of movement, and I'm going to now do the following as well. I'm going to simply fast forward a bit, and go backwards. And when you see that, as it kind of grows, So this is actually a nice one here. Okay, so I'm going to put the arrow right here at the tip. And then after approximately 45 minutes, look what happens. It will actually grow. I'm going to go back and forth so that you can actually see the cells growing. So you can actually observe this directly under the microscope. Here's a little bit bigger. Okay, same here. Again, let's put it here to the tip. And then you can actually, let's go back and forth again. So I'm kind of also reversing, because I can scrub the video, So you can, and you can actually observe now the growth of these cells in time lapse. So, and I think it's kind of cool. Just it's from an educational perspective. After all, I'm a teacher as well, and I want to have also material that I can show to the students. Yeah. Could you not just press the affected area of the orange onto the paper in the Petri dish? Yes, of course you can do that. The reason why I've done it like this with so the question is, can you just take the whole thing and just press it? Where's the, just a second, desk. The desk. Here it is. Yeah, no, that's the other one. I'm too big here. I've got so many, yeah, that's the better one. So could I not just simply press it on here? Yeah, of course you can do that. But what I wanted to demonstrate a little bit is there where you've got how do you say, the toothpick, yeah, then you can, this is basically the places where you can actually see, oops, now I touched it, the streaks, yeah, that's all, yeah, of course you can directly transfer it as well. You know what, why not try it? Who cares really? You can do something like this, yeah. And then, yeah, you're going to transfer quite a lot of these spores directly on here. Yeah? So let's do the following now. Later on, I also, of course, want to put the strawberry and all these things under the microscope. Yeah, side issue. I did. Some people were asking me. These are not new ones. It's a little bit dirty still. I tried. I cleaned those, so I'm reusing those. And I ordered myself a, how do you call it, an ultrasound cleaner. Unfortunately, I had to send it back to Amazon because it was leaking. It was a manufacturing mistake. Because some people want to know how am I cleaning those slides, and yeah, I washed them, but it's possible also to use, or at least I'm experimenting also with ultrasound cleaners. Yeah. So what I need is I do need a bit of water. Okay, for making a mountain. Maybe this is not the best jar. It's a little too big, it doesn't matter. And then I will simply put a bit of this water. You know how to make a, simple microscope slide. Yeah? Let's put this away so that it doesn't tip over. And now we have to kind of transfer this. And look, I'm a little bit hesitant here. This is my favorite tweezer because it's an all-purpose tweezer. You can actually also use it to transfer water. Really versatile. However, it does have one huge disadvantage. It's very pointy. Okay, so you have to be a little bit careful with that, especially when you're picking up possibly infectious material, right? And then you could injure yourself. So I'm just saying, right? Because the safety aspect was already mentioned. And now what I have to do is, and I'm gonna make it bigger here, and yeah, there is actually quite a bit of fungus going on here, and it's not quite as easily visible on video. Yeah? But let's simply pick up some of this stuff here and transfer it over into water. If it only goes off. Yeah, here. Now we go, and a cover glass goes on top. Okay. So, and it's actually a whole colony that I've got here. Yeah. It's probably a little bit too dense, but let's have a look at it under the microscope. I have to change over. And where's the scope? Where's the scope here? Ah, yeah, here, a little brighter. Ah, yeah. That's at low magnification. It's way too dense. I'm going to try the whole thing again because it's simply way too dense. I should have spread it apart a little bit. Oh, forget it. Even an air bubble here. No, bad. Yeah, here a bit, here a little bit on the side. Let's go up. You hear these are basically the cells of the fungus. Yeah, those string-like extensions that you see. It's a very dense network. So, and here we go. Yeah, and as we're enjoying the look of those cells here, I'm going to read again a couple of the comments. Okay, so again, very much thank you very much for the donation. Okay, so maybe maybe he's choosing the white and not the gray. Is the white area another mold than the gray area, or yeah? So that's the question. Okay, so the question is the following. Let's here. in the orange, right, there is this gray area in the center. These are the places where the spores started to form. The mold is growing outwards, right? And so basically new mold is white. And there where the mold is already a little bit older, the green gray area, that is where you find a lot of spores. Yeah? So you see that just a couple of hours ago or today in the morning, the gray area wasn't quite as the gray area here wasn't quite as large because it did not form a lot of spores yet. Yeah? So that's the thing. Yeah. I was going to buy an ultrasonic cleaner. Yeah, I bought an ultrasonic cleaner, and again, there was a manufacturing mistake. Yeah, so this is a little bit how it looks like here, but I'm going to make another slide, right? Because this is not the one. Yeah, it's not quite as nice looking as I was hoping for. So I'm going to add again a little bit of water here, desk view, and then we're going to have a look. Yeah, here we go. Okay, number one, and maybe I'm going to take something from here. Carefully scratch it off. Sometimes it's difficult because it doesn't want to get off quite as easily. Here, let's do the following. Yeah. So normally, of course, you grow things like this on agar, nutrient agar. Yeah. Generally, I have to be honest with you. The whole thing with growth of microorganisms is such a thing. You know, you got to be always a little bit careful with these. Generally, because sometimes you don't know what you're growing. So here we go, and let's have a look at this here. Okay. I'm just going to do the following. I'm just going to take it out as it is. And the scope view again directly, and I'm just going to directly put it in here. Here we go. It is of course much better, right? Yeah, and you can also see some of those dots that you see floating by. These are of course spores. So yeah, and yeah, that is basically penicillium. So I think it's the same genus as the fungus that's growing on camembert cheese, right? Yeah. So what you have over here, for example, it's called digitatum because of these finger-like extensions, like the digits, of a finger, right? And at the ends you have the spores forming. Yeah? So that's kind of very typical of this fungus. Yeah. And at the ends, then you have the spores forming. Yeah. So that's basically in DIC. Some people want to know how does this look in bright field, because most of you probably don't, quite certainly many of you don't have a differential interference contrast. So I'm going to put away the filter. This is how it looks in, close this here. This is how it looks in real colors, in normal colors. Okay, so that's unless you want to use some filters. And let's try the following. This is now 60 times. So you can see it's quite a bit larger. Let's put in this here again. Okay, you see it takes away a lot. And then let's also have a look at phase contrast. Yeah, this digitatus plus milk is cheese. I wouldn't say that. No, because the species is different. Yeah, they're using penicillium is just a genus, and there are many species. Yeah, so you always have to be very careful that you don't just use any type of mold, but you have to make sure that the mold is actually of the correct type. Yeah? So that is now that's now phase contrast. Just in case you want to know. It's probably not the nicest. Phase contrast is not always suitable, right? It's very good for observing bacteria, but usually thicker specimens not quite as good. Yeah. Okay. So now let's put the fungus in the petri dish with the bacteria. Let's replicate Fleming's mishap. Yes, you'll be surprised. I actually have been thinking about this as well. The thing is the following: a little bit of biology, because Alexander Fleming. Well, actually, there are now discussions going on. Apparently, the Scottish surgeon Joseph Lister, he has already been using fungi for yeah, in band-aids and so on to treat infections. But apparently he did not document that in any publication. But Alexander Fleming, the Scottish scientist, back in 1927, discovered the following, and that's actually quite an interesting and relevant thing here, and that's why it's called penicillin. The antibiotic is called like that. This is because in one of his petri dishes he was actually growing bacteria. And then he saw that there was a contaminant, a colony of a fungus growing, right? And next to the colony, there were no bacteria growing. So he kind of concluded that maybe the fungus is producing a substance that kills the bacteria. This was actually the case. And that's how penicillin was discovered. And then they started to grow the penicillin in large amounts for medical purposes. Yeah? So this is indeed, the case, but let's have a look again at the scope view, and yeah, let's do now the following. And yeah, it's a little bit. We have to close the condenser here. Yeah, wrong. So too bright. So now what I want to do now is maybe I don't know. Maybe let's try a little bit of staining just for the fun of it. Then I want to put also some of the fungus of the tomato and the strawberry under the microscope, and then yeah, that's the thing that I kind of want to try. Let's do the following. Let's try to find this here again. Yeah, so that's the let's do the following. I've got. Or maybe I should be staining it first. Maybe that's easier this way, because if I just put the stain directly on it, then maybe. Yeah. So the standard stain. I've got 3 stains here: saffronin, crystal violet, which is a very powerful stain, and Löffler's methylene blue. I want to try saffronin. I don't know if it actually does stain or not. So what I'm going to do is, why not try all of them? Who cares? We've got plenty of fungi. So let's put here. Yeah. So I'm going to do now the following. Okay. So that's the saffronin solution. Remember, maybe some of you remember from previous weeks I've been experimenting with that as well. And I'm going to try to grab a colony. Let's take this colony over here and put it in here. And let's spread it apart a little bit. Hoping that I'm not losing it. It's still here. What I'm doing now is I'm actually removing the stain again, because I want to wash it, the whole thing. I should have maybe changed the tip here as well. I want to add a little bit of water, because otherwise, if not, then there is the possibility indeed that it's going to be too dark. Okay. Yeah, I can't go into the water, so maybe I should have a separate pipette just for transferring water. Okay, but I think that's fairly bright. So, as a matter of fact, also for me the first time to see if saffronin. You almost barely didn't see that because it was hidden behind my picture here, my video. So, this is so basically a drop of saffronin and then washed. And let's put it in here and see if it has stained cells. Of course, every time when you do that, you have to be aware that the staining might actually kill the cells. So I have to find it. Ah, here it is. Okay. Actually, it seems to have stained quite nicely. See, it's still very dense. Here, maybe that's a little bit better here. Okay, let's go up. Oh yeah, actually it does look. So by the way, that's bright field now. Okay, so 60 times. Actually, I'm quite happy with the results here. Yeah, it looks nice. Yeah. So so the saffronin really seems to have stained it quite well. Yeah, so I'm pretty happy. So saffronin is also used as a counter stain for the gram stain for when you're staining bacteria. You can see that, yeah, seems to have absorbed the pigment quite well. And as I've shown you, if you want to do that, then you have to remove again you have to wash it, right? You have to remove all of the excess stains so that yeah, you get a nice contrast. Yeah. So I'm reading again here. I'm reading. The crystal violet from the previous live stream is still visible. Uh-huh. It's unfortunate that the section of the tardigrades is so short. Okay. Try putting some in a small vial and shaking it rapidly to loosen them up or to distribute them with the water. That's actually possible as well. Yeah, kind of you collect some of this and then you kind of shake it and then you see how well it actually yeah, separates. Yeah. Oh my gosh. Dave Lewis, did you receive the frog spawn initial cell separation? Yes, I did. And I forgot to prepare it for the live stream. Heavens. What am I going to do now? There was a really... Dave Lewis sent me another video to be shown where you see the actually initial cells of a frog egg dividing. And I wanted to show that to you, but it's on my other computer. You know what I'll do is I'll show it to you next week. Okay, this is. But thank you very much to Dave. It was really a fascinating video, and if you allow me, I'm also going to show it to my students in school because it actually shows an early embryonic development from a fertilized frog egg, and then it's dividing, and it's really fascinating. So thank you again. Yeah. So in polarized light, well, it's not standard polarized light what I have. It's a DIC, which does use polarized light, but it will also introduce our artificial colors. So that's kind of DIC now, and I can change. I can change now using a prism. I can change now also the background color. So this is yeah. So this is. So yeah, again, thank you very much to Dave. Actually, I wanted to do that, and I watched the video and was really happy because it was really fascinating. But yeah, so so that's basically a combination of DIC and staining. And you know what? Also something I did not try. I'm going to use now phase contrast. I'm going to use phase contrast and staining. I don't know how well this actually works. This is. So that's now phase contrast and staining. That's also interesting. Okay, also looks kind of okay. Yeah. So just for the fun of it, I'd like to try the other stains as well. Honestly, I have to admit, I have not tried that yet. So one of the things that it's a little bit become a little bit of the way that I'm doing these live streams is that sometimes I don't even know what to expect myself. So I'm going to now do two of them. Here, the left one here is crystal violet. Okay, which I might also have to wash a little bit because it's such an intense stain. And this one here is a ready-to-use solution which I do not have to wash. It's Löffler's. Löffler's is a basic, an alkaline solution containing methylene blue and alcohol. Yeah, and yeah, I'm just going to try this again here. Yeah, I'm going to try to scratch off some of the fungus and put it in here. I think I have to now clean the how do you say, the tweezers, and I'm going to try another one over here, scratching it off a little bit. Yeah, there's something on here. Yeah, so I can directly put a cover glass on the methylene blue one because it's not it's already a little bit diluted, it's ready to be used, and it's not quite as dark. Therefore, the crystal violet is kind of very intense. So what I have to do with the crystal violet is I have to wash it. Okay, so I'm going to add a little bit of yeah. I'm going to add a little bit of water, and I think there's still something left here. Okay, and I'm going to, you know what, for the water I'm going to use just the other pipette from now on, because otherwise I always have to keep on changing it. Okay, and now I can remove all of the excess. Okay. And cover glass as well. Cover glass as well. Oh yeah, it's still staining quite intensively. So crystal violet on the left side. Crystal violet on the left side. Let's start at the very low again. Crystal violet at the left side. And where is this? The scope view. Here we go. So that is, it's not a lot. Okay. There's some more. Here's more. Here. Yeah. So that's the crystal violet. That's still super dark. That's the crystal violet. You see how dark the background becomes. So let's 40 times. Yeah, and focus. I don't know. I kind of like the saffron in more. It was not quite as, because it's. I should have washed it a little bit more maybe. But the crystal violet here stains quite nicely, and you see the background is still quite blue because of all of the pigment that's coming out. And now let's try the other one over here. That's the methylene blue. Does not seem to absorb the pigment quite as much. So that is also illustrates a little bit on how different the different chemicals actually react. Here a little bit more. I love experimenting around. Yeah. Can you show us in the future live stream how to do a gram stain? Yeah, I'd have to grow a few bacteria first. I do have E. coli bacteria. Generally, it would be nice to have some gram-positive bacteria, and I'm thinking about maybe getting some bacteria from yogurt, because yogurt does contain gram-positive bacteria, and I want to use safe bacteria. I don't want to grow, it's really easy to grow bacteria from your own skin and so on, but I don't want to be a bad, role model, because strictly speaking, you shouldn't be growing your own bacteria at home unless you know what you're doing. There are even some legal constraints. Strictly speaking, you are in safety category number 2. It's the second highest biosafety hazard level if you're working with unknown bacteria, because you don't know what you're growing. So this is kind of the methylene blue one. Okay. So I'm reading again the comments. So I'm reading the comments. Okay. So far, that's it. Okay. Now what? I do want to do the following. Something I have not done yet is the following. I want to also show try to look for some spores directly. And for this, I have to go directly to the orange. So let's put the thing here. You see over here, they are not quite gray yet. So maybe the sample is still too fresh. By the way, if you see that the whole thing is kind of drying out already, so all you do is you add a little bit of water here on the side, and you allow the filter paper to kind of do the rest. Yeah. So you don't want the whole thing to float in water, but it should be moist. So for actually observing some spores, I would actually pick something off from the green area. Okay. Some of my, and here we go. Same story again. A little bit of water. Well, it's probably a little too much, but who cares? Yeah, you can either maybe use some toothpicks. I've already removed quite a few of them here. And I found that many of these spores are actually kind of a little bit hydrophobic. So it kind of repel water a little bit. So that's it. Just throw those away again. And of course, it would be kind of nice to not only observe the spores here, but maybe add some orange juice and then kind of wait a couple of hours to see if it actually starts to grow from the spores. So And where are we again? The scope view. It's way too bright. Here we go. Ooh, lots of stuff here. And a little bit out of focus. Okay. Here, these are the spores, right? So that's the stuff that's causing the allergies if inhaled too much. Yeah, so that's again with a little bit of color. And let's make it, you can change colors here as well. Yeah, so basically, that's basically the thing that causes the problems, because when it spreads, it settles down on new fruit, then yeah, you have a new fungus growing. So far no questions. Okay. Yeah, let's have a quick look at the strawberry. I'm also checking the time always a little bit. So let's put the other slides away. Yeah, I've got a container with used slides over there. But we can actually look at the, maybe, I'm keeping the one for the saffron in, simply because it's kind of nice to look at. You know, so that's unbelievable. Look how many there are. Unbelievable. That looks quite nice. Yeah. So let's have. Oh, maybe you know what? I think maybe that's actually good in phase. Let's have a look. Yeah, no, that's not phase contrast. Yeah, the spores look nice. Okay. Yeah, maybe you see actually some of them are kind of shaking around a little bit. Yeah, that's the Brownian motion, so they're not moving on their own. Yeah, but that's the shaky movement is Brownian motion. Are the spores the same size as yeast cells? I think these spores actually are significantly smaller. No, why? Why think? Why do I say I think the spores are? I have over here some yeast, so why not try it out? Okay, I mean that's actually the whole point of a live stream is to as far as I can do that, at least try to fulfill the requests of the community. So I've got some yeast here. Why do I have some yeast here? Because I'm using the yeast to feed my water fleas. Okay, and try to do following. I'm going to improvise. I found also out that sometimes the yeast is does not dissolve easily. So what we have here is these are now that's dried yeast, and I have to dissolve it first because the individual cells are clumped together. Okay, so let's do it like this. Okay, and let's compare now the size between the spores and the yeast as requested. So let's have a look here again. I'm going to let's use the 20 times the scope view. Here, this one here, this one here, this one here. It's always too bright here. So that is these are the spores. Okay. Let's just remember how big they are, and these are the yeast cells. They're pretty much the same size. Yeah. So these are yeast cells. Let's go up. These are the yeast cells. Actually, they almost look very indistinguishable. Yeast cells and spores. Okay. Spores and yeast cells. Okay. Yeah, so I think the spores do look a little bit more regular than maybe the yeast. And here again the spores. Yeah, so. Yeah, I don't know. Yeah, okay, but I think, yeah, thank you for the question. This seems to answer it. They seem to be fairly similar in size. Yeah. What magnification? Yeah, you can actually, the magnification question, you see those, that's one of the reasons why I actually included this. Unfortunately, you cannot always see the magnification. What I have is the following. I have over here, it's this one over here is 40 times. I've got now the, yeah, So that's 4 times, so 40 times, yeah, 4 times, 10 times, 20 times, 40 times, 60 times is already touching the the microscope, yeah, the cover glass. Yeah, so that's a little bit the problem here. But let's try the 60 times over here because I think there's a little bit less water. Here we go. So that here is now 60 times. Yeah? And in bright field. Yeah? I'm not going to use the 100th time. So that's the yeast. Okay. Are the spores spread in the air? That's the question. And the answer is yes, that's the whole point. That's the whole point actually of spores. Unfortunately, I want to add, because that's how fungus spreads, right? And it's spread over the air, then it settles down a new place, and then it starts to grow again. Yeah, and that actually, because the spores are spread over the air, that's actually a problem for some people, because when they inhale it, then they might have some allergic reactions or asthma and so on. So it's similar to pollen grains. Yeah. So that's a little bit the thing here. So we now do the next thing. I want to look at the strawberry. Also not looked at it yet. Okay. And let's have a look here. Same procedure as always. And let's add a little bit of water again. Here we go. I need to clean the tip here a little bit because I do not want to have any significant cross-contamination. Not that this is a huge problem, but I just want to make sure that when you look at something, it's really only one type of thing that I'm looking at. So, and let's try to carefully. Yeah. This actually goes off much easier. Lots of fuzz here. Yeah, as soon as it touches the water, it doesn't want to go off. So let's do the following. Let's carefully, yep, here we go. I find that sometimes the mold is fairly, how do you say, hydrophobic. Yeah, it's kind of like it repels water quite a bit, interestingly. So it is more than enough here. And here we go. So, and now again, under the microscope, so that's the strawberry fungus. And let's have a look at this. Start, no, where's this? I have to go to scope view here. So that's the strawberry fungus. It's too bright. Let's go up with the magnification a little bit here. Yeah, that's the strawberry fungus. Okay, let's add a little bit of color again. I mean, it does look a bit different. Yeah. And I know I know I'm kind of repeating myself a little bit because I've already said it so often, but maybe some of you, yeah, were not around yet. But one of the very typical things of a fungus is that actually it's branched, right? Yeah, so you see over here the branching here. And that is something that you see in fungi, but not in bacteria so much. Okay, but there is a small difference that I do want to talk about possibly, but I have to go up a little bit with the magnification. Ah, yeah. So this seems to be an aseptate fungus. I cannot use the high magnification now. No, actually it's a here we see those. Okay. Interesting. The thing is the following. Sometimes you can actually see the individual fungal cells, but in this case, they're also separated from each other. So you see that they're actually, so it's a septate fungus as well. You see over here between the cells here, right? There are cross divisions. And there are different types of fungi. Some fungi don't have those cross divisions. Then they are referred to as the so-called the aseptate fungi. But this one over here has these cross divisions. Yeah. So, and if you're now very patient, what I would then do is which I'm not going to do now, but if I take a video of this, then maybe it's possible to actually see how it grows, right? And that's what I've been doing before. Yeah. So, I'll answer and comment on some of the questions that you've got later on. Yeah. Demodex, somebody's trying to catch face mites. I'm planning again another face mite video myself. Yeah. So that's basically the strawberry fungus, okay? And let's, for the fun of it, what time is it? Okay. Desk view, let's have a quick look at the fungus from the tomato. Okay. Here we go. And you know what? Because I just liked it so much, let's try to do a little bit of staining again. Add a bit of the, yeah, and this also goes off much easier. But it goes off much easier from the tomato, but not from the tweezers. I need another toothpick here. And now it's on the toothpick. It's all the way on the bottom here. Yeah, and it's too little. Maybe I need more. That's a lot. You cannot see it again. It's difficult. It's the fungal cells, they grow into the substrate, in this case into the tomato. And it takes patience. Here we go. There is now a little bit here. I'm going to remove the excess again. Okay. And we're just going to drop a cover glass directly on top here. I mean, it looks like blood. It's not. So this up, here we go. But it kind of looks a little bit like it did not stain quite as much as, well, we're going to have a look. Okay, let's have a look how this looks like. And so here we go again. And where is the scope view? I see it's way too, yeah, this is basically what happens when you don't remove the stain. Yeah, too dark. No, not enough contrast. Okay, so I mean, it did stain it obviously, but the point of staining is that you see the cells at a different color than the surrounding. Ear bubble, ear bubble. Yeah. But over here, maybe see, these are the cells in ear, and they have kind of passed on the pigment. Yeah, that's also okay now. Yeah. So it's interesting. These cells here are in ear, and evidently, they're. I mean, this is kind of also nice because they seem to be obviously they are connected, and this means that once the stain has been taken, well, but maybe the ear bubble formed afterwards. Yeah, but it doesn't matter. In any case, you see that yeah, it does stain quite nicely as well. Yeah, modern art. Okay. So there are a couple of off-topic questions that I would like to address later. What else do I want to do? Yeah. Then over here, the strawberry we looked at. You know what I would like to try is, yeah, I'm going to go back here again. Yeah. So I'm going to go back to the desk view. I'm going to remove those slides now. And I would like to now do the form. Let's put them somewhere over here into the trash container. I have a separate trash container as well. And yeah, what I would like to do now is the following. I just want to show you very briefly another type of mold, which is not a mold, but simply to keep you a little bit updated. These are the slime molds, and I nearly killed the specimen because I forgot to add some water, and now it started to dry out. But I put some oats on here, so I hope that it's going to regenerate. Huh? So just for your info. However, there is one thing that I do want to show you, because some of those oats here are quite slimy. And I would like to show you also a little bit, because we now looked at cells of fungi, but I kind of expect there to be quite a few bacteria. Okay, so I'm going to do that now. I'm going to first show you under the stereo microscope. Where is it? When does the video come? You see all of this slime here that you see overgrowing here. It's not slime mold. Okay, the slime mold is this one over here. Let me put this over here, right? The slime mold is the stuff over here, and it is not a mold, yeah. And this one here, the slime that you see over here, I'm pretty sure it's not slime mold, probably bacteria, right? So, and I don't know which ones, so one has to be really careful. But I just want to also show you a little bit how bacteria now look like, in comparison, in case you have not seen that, yeah, because they look quite different from fungi. So again, the desk view. I just want to give this to you as a little reference here. And drop of water. Now I do have to be a bit careful. I'm going to take some of this slimy. Let's go down. And I suppose that there are quite a few bacteria on here. This is already a lot, okay? This is probably already a lot. So, then I throw it away and I put a cover glass on top and let's have a look. Scope. Okay, there's those dots that you see here are too large for bacteria. Okay. No, that's a fungus as well. Those oval things that you see, these are fungal conidia, but those dots in the background, those tiny dots in the background, these are actually bacteria. But these bacteria are kind of a little bit smaller than the other ones I've seen. So, yeah. Okay, it's not a pure culture in any case. So what you see over here, very briefly, those oval things that you see here, they look very much like fungal spores, like so-called conidia. And those dots in the background, these are bacteria. Yeah. So you see a little bit, and I'm right now at my 60 times objective. So you see how much small bacteria actually are. But let's do the following. Let's go 40 times phase. 40 times phase. Yeah, and that's very typical. Bacteria are those tiny black dots that you see in the background. So you see, that's a little bit. That's a contaminant on my plate, right? Therefore it's not ideal actually, because those slime molds they don't. I've seen they don't like. To be around. If there are too many of those bacteria, then they try to avoid them because the bacteria also produce a lot of waste products and so on. And generally, the slime molds, they always like to be in a place where there's the fresh cereal. So let's have a look. Okay. Why not lift the cover glass and rinse it with water? Okay, let's do the following. Let's try that. Okay, this was, I think, a question referring back to the previous one, which was over here. So I'm going to now do the following. I'm going to look, just so that you see what I'm doing right now. Yeah, over here, this is there's a little bit of water here on the side. But what I should do is I should actually be careful, and I'm going to add some alcohol later on here, simply to kill off those bacteria. So let's put them away. So the question was as follows: if the safranin is staining too dark, okay, like, oh, thank you very much. The questions are here. So again, over one page. Let's try that, and let's try to improvise a little bit here. Let's try to lift carefully the cover glass. The fungus is now sticking on the cover glass. So I'm going to try to do it like this. Carefully removing any excess. If there is nothing on here. So this means I can do the following. I can simply remove the stain completely. And I've got it over here. So I'm going to try the following. I'm going to try to simply... Now this is now a little bit improvised here. Let's try to put this down. Yeah. Yeah, this is better. And cover glass. I'm going to remove the saffron and stain from the cover. And I don't even remember anymore which one was this actually. Was this the strawberry or the tomato? So let's remove all of the excess. And yeah, I think this should actually give us better contrast. So, the scope view. This I'm still in the phase contrast. I'm still in the phase contrast one. Yeah, turn down. This is actually also. Ooh, this is much better. This is much better. Actually, this looks pretty. This actually looks quite nice. Okay. So, here we go. Yeah, this actually looks quite decent. So that's saffron and stained, and now because there was the request, I removed I lifted the cover glass and kind of washed everything again. It looks quite okay. Yeah, looks quite okay. So what I'm going to do now, for those of you who just joined in, I'm going to do a little 1-minute recap. Okay, for those of you just joined in a little bit later, because the main point of the video was actually to show you how I've prepared those petri dishes to grow fungi. So I'm going to I decided to quickly do that again, simply because it's also on the on the thumbnail and also in the description and so on. So in order to make yourself a simple paper-based medium, a growth medium, you do the following. You just take a petri dish, you take a pencil or a marker, okay, quickly mark the whole thing so that you know a little bit something about the size, and then you take scissors and you generously, and then after I've done this, I'm going to talk about the questions that you've sent. So I'm going to do this very quickly now. You generously cut out the filter paper. So I'm just using 2 of them. Okay, I put them in here. And now I'm going to do it in a very quick way. I add a little bit of this freshly pressed orange juice here. Okay, I allow the whole thing to spread out a little bit. And it's just like this. So it's a little bit. It will take a little bit of time for the liquid to distribute. Okay, just remove the. And once you have done that, what you can do is you take a toothpick. Okay, and you simply collect some of the spores from here, rotating it. You see how the toothpick's tip actually turns gray. Yeah, these are all the spores. And you then do the following. You either transfer it directly here, and then you can actually see that it starts growing from there where you actually streaked the toothpick, or you can do it in a different way. Just as suggested, you take the orange and you simply roll it like this, transfer the spores, and then you can pick some of those growing cells and put it under the microscope, and then you're able to hopefully see a time-lapse video like I've made before here. So this is again a time-lapse video, and over here, if I actually scrub ahead, then you can actually see that especially the top one is kind of moving and growing. So this was actually over the course of about 45 minutes. It loses focus a little bit. But you can see those. So it's basically on the petri dish a little bit of a controlled environment, so that you're able to observe the cells better. That's it. So let me have a look here, the desk view. So this was simply something that I very quickly that I've done, so that in case you wanted to know how I've done this. Is there much difference between lab grade filter paper and regular coffee filter paper? I mean, if let's put it this way, I don't know if there's a huge difference. Lab grade filter paper has one significant advantage: it's standardized. Okay, I would say I think the filter paper in the lab that I'm used to is actually a little bit thicker than the coffee filter paper, but honestly, it's got to do its work, and ultimately, for practical purposes, probably doesn't matter a lot. And maybe even I've seen some very thick lab filter paper, so I suppose you can actually also buy them in different thicknesses for different purposes, right? Maybe some filter paper for paper chromatography is again different than maybe filter paper that is used simply for filtering. Yeah, but my philosophy is a little bit is to use, if possible, those materials and the equipment which is readily available. And of course, it's easy for me to simply go online and order a whole pack of standard filter paper. That would be the normal thing to do. But I decided simply to just use coffee filter because that's readily available, and many hobbyists are probably going to use those. Yeah, and it works, as you can see. So, but I would say it's a question of experimentation. Yeah. Yeah, tolerance level of sterility could be sterility. It's not sterile anyway, I would say. So if you want to use in a laboratory setting something like this, then you'd probably have to sterilize it in any case. Yeah, because yeah. So having said that, what time is it? Okay, I'm now going to spend a couple of minutes, like every time. Okay, I'm gonna first different thing. In the YouTube channel, what I'm doing, I've started just recently. I'm trying to also do Q&A sessions and something like a little bit like a podcast for about half an hour once a week. You can find it actually also in this channel over here. I'm just experimenting a little bit, and I'm going to also address some of the questions that the viewers have. So that's one thing. Then live stream, I occasionally make some tutorial videos, but it's something that's very important, especially in the last couple of weeks and months. The number of views of these channels have gone down, right? Quite dramatically. I mean, that could be also a seasonal change, maybe, yeah, depending on the season and so on. Yeah, if you have any specific suggestions on what you want to watch or whatever, I'm very much open, or what I could improve. I'm very much open for feedback. I've been also experimenting around with doing scripted videos, which I personally don't like so much because it's kind of read off and it doesn't quite sound quite as natural. But interestingly, those scripted videos get more views. So maybe people like scripted videos more because more of them watch it. I don't know. It's kind of, for me, it's a little bit counterintuitive because, I'm in any case, I'm happy for any kind of feedback. I'm personally doing this also because for fun, and it interests me, and because I'm a biologist. I also use those videos a little bit for my biology lessons. So I have this general, fun doing things like this, so I don't mind so much. But if you have any specific recommendations or requests or whatever, I'm very open. Okay. Will microscopic mysteries ever return? That's a good one. I've got several microscopy YouTube channels, and one of the YouTube channels that I've been experimenting around with I called Microscopic Mysteries, which is focused on stereo microscopy, where basically I'm showing you things, and then you, in the form of a YouTube short or a short video, and then you're supposed to guess what it is. Okay, so this is one of the things basically, will it ever return? I don't know. It doesn't take a lot of effort making those videos. It's more a question is, what does the community want to see, right? And so for this reason, I've been experimenting around with a lot of different formats and styles. And I've got this other channel called All Things Microbes, which has very few videos where I'm trying to do more biology things. So you see, I've got a whole bunch of projects running. But that's actually the main channel that I have, right? So this is, I think, more about living things. It's also one thing I've been thinking about. Thank you very much for this comment. Maybe I should place a stronger focus on living microorganisms as such, and not so much on other I would say, cells or fungi. Also, that has been something I've been thinking. People requested me to do staining videos. So that's all why I've been also doing a lot of staining in the previous videos. And this is yeah. So I'm quite open here, but I'm very happy for general feedback. Yeah. Many people like scripting videos because they can see it silently at work. Wow. Again, people like scripted videos because they can watch them during work. I never considered this possibility. So basically, people are actually simply listening to the video, the scripted video, like in a documentary type format, and to get the info. Thank you very much for this info. I've never considered this possibility. That people might actually watch the videos primarily for the sound, but that is really important as well, because then I know that I can also make videos that people can listen to, and I will place a stronger focus maybe on simply on the text, on the audio, and not so much on showcasing things. But this is a very visual format here, obviously, because I'm showing you the things. But maybe that's maybe scripted videos are or semi-scripted videos allow you to also watch the things without actually watching them, because you listen to them. Yeah. So I'm going to go through some of your questions. Maybe I already answered some of them. Okay. What are the safety concerns on growing such molds cultures like this at home? Yeah. Honestly, be careful with molds. However, I did. Do a little bit of research, and especially those molds growing on citrus fruits, oranges, lemons, are not known to be extremely dangerous to humans. However, having said that, they do scatter spores and they can spoil food, and you don't want to inhale the spores in any case. Yeah. Have a dead spider? Do you have any tips on observing the spider? Yeah. Generally, dead spiders and other small animals are generally too large. Yeah. So what you have to do, and I know this sounds a little bit, you gotta pick it apart and look at the legs. Or what if you want to keep the spider intact as a whole, what you do is you put the spider on without cover glass. Okay. On a microscope slide, spider goes on top here. Maybe you want to use a little bit of glue maybe to fix it. Yeah. Just on the bottom side so that it doesn't fall off. You put it under the microscope. And with 4 times, with a low power, you shine a flashlight on top. So that actually does work. Yeah. So this is actually a possibility of seeing opaque specimens. So thank you very much for the donation. Okay. Generally, for your info, I'm using the donations and also from the monetization of the videos. I'm using this to actually buy equipment for microscopy. For example, I bought myself recently a box of slides that I did not show you yet, which contains basically cross sections of human tissue, which is it's not a nice sample of tumors, cancers, and so on. I have only the problem I don't know what to say to those, because I'm not so much trained in medical histology. So this means if I see a cross section of a tumor of a certain tissue, I cannot really tell you how it's different from a healthy one, right? So I still have to learn a little bit more to actually make sense of the things that I actually see under the microscope myself. Yeah, but just for your info, I'm actually using the donations to buy slides. Yeah, couldn't you just press the affected area? I've done this. Yes, that's indeed possible. Okay, is the white area another mold in the gray area or in all the spots? So I also answered that. The question here again is you see if you have a mold like this, then you see that there are parts here in the center that are dark green or grayish, and these are basically the ones that contain the spores, and then the white part is the one which does not yet contain spores, and the fungus will grow around here. And look, if you actually look at, oh my gosh, thank you very much, another donation. And you see already over here on the other side you also have fungus developing, right? So it either could be that maybe the spores have spread on the outside, or maybe the mycelium maybe has already reached the other end here on the inside of the orange, right? And then started. I don't know, but essentially what the fungus does is it secretes enzymes which digests the surrounding, and then it absorbs the nutrients. Yeah, and that's essentially the reason why spoiled food should not be eaten, because even cutting it away, the fungus is not a good idea, because the inside actually contains then the mycotoxins. Yeah. Again, a big thank you to Peter. Okay, thank you very much. Okay, so yeah, so ultrasonic cleaner, yes. Honestly, I'll be honest with you, it's already the second ultrasonic cleaner that I'm sending back. The first one didn't even turn on. I sent it back and I bought a second one now just a few days ago, which is actually quite nice. But it was really the inside container was leaking. There was a small hole in there. It was a manufacturing mistake. And that's something you cannot have. So it also goes back. My favorite thing to clean my slides at work at Alconox and Kim wipes. Okay, yes. Yeah, there are different ways how you can do that. I've also found for cleaning slides, I've also found dishwashing powder to be quite effective. A router for just photobombed my amoeba time lapse. Okay, yeah. Usually that's in a natural water sample. You always have things floating around and swimming around. Yeah. How do you know it's a penicillium fungus? Okay, that's a good one. That's Penicillium digitatum is the fungus that's commonly found on citrus fruits. Okay, and I assume so. Therefore, I assume it's Penicillium digitatum. I know that from simply the shape of the mycelium, it looks like that. But I know that maybe mycologists are probably going to say, and rightly so. I mean, actually, strictly speaking, you have to do a DNA test. Just because certain organisms look similar under the microscope does not necessarily mean that they're related, right? And concerning that the biodiversity is so large, yeah. So I get your point here, right? And the way that I basically said it's Penicillium digitatus is simply because it grows on the orange, right? And it kind of looks like it. I get it. This is a very, very weak argument. Okay. Yeah, I have to agree with you here, right? Now put the fungus in a petri dish with bacteria. Let's replicate Fleming's mishap. Yes, in order to do that, I'd actually have to grow the bacteria properly on an agar petri dish, okay? Because on the filter paper, I don't think that you're able to see the colonies properly. Try putting some in a small vial and shaking it. That's also, of course, a possibility. You can try to shake it, or there are these laboratory machines that essentially will shake a tube for you very quickly. Yeah. Did you receive the frog spawn? Yes, I received it. I already talked about that. Thank you very much. Unfortunately, I totally forgot about it. Kind of preparing the video, and it's not on my other computer. Yeah. Polarized light. Yes, I've done this as well. Having said that, however, my polarization setup is a little bit different than maybe yours, because I've got also those Wollaston prisms in there, which are all... Thank you very much. I cannot bear trees now is also donating. Thank you very much. Okay. Yeah, so that's polarized light. You can try to use... I'll be honest with you, I don't have, on my microscope, I don't have standard cross polarization. I've got this DIC setup which contains all of these prisms and so on. But if you want to try that, you can simply you simply take some polarization filters and put the slide between two crossed polarizers, and that works. Yeah. To do a gram stain, working on it, I need bacteria for that, and specifically I need gram-positive bacteria to actually show you how it actually works. Okay, the gram stain, I have to tell you, is somewhat a more traditional stain. Yeah, it's yeah. In a stable aquarium, where should the ammonium nitrate there should be ammonium and nitrate consuming bacteria? Is there a way to detect them using staining and a microscope? Yes, long time ago, over 20 years ago, I used to have an aquarium at home. I was actually quite fascinated by it. And a proper bacterial balance is extremely important, and usually those bacteria are found in the filter. Of course, not only bacteria, they will grow in the sediment. That's why you have the gravel and all these things. But the filter contains a large surface area, and you have to have those bacteria also in there, because if there is so-called nitrite being formed, which is kind of poisonous, then it's converted by those bacteria to nitrate, which is then less poisonous, right? And also the waste the fish produce. So what you have to do is you have to actually take some of the stuff from the filter and put it under the microscope. However, And that's a general problem with microscopy and bacteria. Just because you see bacteria, you don't know which ones they are that are actually doing the conversion. You just see bacteria, right? And you can be quite sure that many of them will actually do this metabolic process, but there is no way that you can actually see this process under the microscope. Yeah, it's itself. So if you see 1,000 bacteria, you cannot just by looking at them say which ones actually will do that. Yeah, so that's a little bit the thing. So you see, as important as microscopes are, there is of course a certain limit. Yeah, the spores of the yeast cells, we found out that they're pretty much the same size. Okay, thank you for showing your samples and the different illuminations. Always interesting. Yes, thank you as well. We're going to do an agar block method in school. So excited. Yes, you can do that, but please be careful. Okay, what you're doing if you're using an agar, if you're making an agar medium with yeast extract, you are growing bacteria at an incredibly high concentration. And I know what some schools like to do because it's pedagogically and from an educational perspective, it's so powerful. What they do is the following. You've got your Petri dishes here with an agar medium. And then what you do is you take the lid off, and then you kind of press your finger into carefully, and on there you kind of roll your finger, like fingerprints, right, on the agar medium. And then you kind of clean the other finger with alcohol, and you do the same one next to it, right? And then you close the lid, you put some film around it, yeah, to close it off, and then you're gonna wait, and then you're going to see, surprise, big surprise, the part with the uncleaned finger, there are lots of colonies growing, and the one where you've actually cleaned the finger with alcohol, there are very few, if no, maybe even no colonies growing, and then this kind of demonstrates that dirty fingers contain a lot of bacteria, and it's very effective, but you should not be growing bacteria from your own skin on a Petri dish. Okay, so that's yeah. So I'm just unless you're really careful, because some students open them up and then they play around with it and then they get the bacteria, and who knows, yeah. And the other thing is if the Petri dishes are not entirely fresh, it could be that there are actually also bacteria contaminants growing on here. Then you're kind of rolling your finger into the contaminants, and maybe just some microcolonies in there, right? So for this reason, how do you say, don't touch the agar medium if possible, right? But I'm just saying this because I know that these are very popular experiments that are commonly done, but I'm a little bit, as a trained microbiologist that I am, who later on then went into education, I'm a little bit, I mean, I wouldn't have a problem doing that because I kind of know a little bit the safety aspects, but you see, there are certain guidelines and regulations of what in different countries of what you should be doing and not doing, right? And yeah, so I'm just saying, yeah. I'm getting sidetracked. I saw your Euromex stereo microscope review. If you had to buy a new stereo microscope, would you consider buying it? Why not? However, Euromex also has other microscopes, and I also made a review of the next year's Zoom microscope, which is a little bit bigger, and which I'm using right now, and it's similar a little bit, but it's a little bit bigger. And I also like the Nexus Zoom. You might have a look at that one as well. So essentially, it's a little bit larger on the bottom. So, but I mean, the company offers many different variations of the microscope. So that means you can actually consider fitting it with different stands and so on. So there are lots of different combinations here. Yeah. The highest magnification that I'm usually working in my live streams is usually 60 times objective. So when looking through the eyepieces, around 600 times maximum. Occasionally, very rarely, I use the 100 times oil immersion objective. The reason is because it's kind of messy, and it's kind of dangerous because when I change the magnification again, there's the danger that I'm rotating the oil immersion objective into the non-oil immersion objectives into oil. So for practical purposes, the 60 is the maximum that I'm using. Yeah, they spread in the air. Yeah. How does the yeast spread? I mean, the yeast that we're using here actually is from a brewer. Usually those the baker's yeast. Yeah, this one over here, this dry baker's yeast, which can be bought, usually is made when making beer. Because when you brew beer, then the yeast reproduces a lot, and then it's harvested. The beer is fermented into alcohol, and then the yeast is sold for making bread, right? So in that sense, it's it's cultivated and grown, right? And yeast can be naturally also found, for example, on fruits and vegetables. They're part of the natural environment, right? But usually not in that high concentration. How does it spread? I guess it depends on the yeast. Maybe also washed away over water, over the air, and so on. Yeah, then it will grow and reproduce maybe when the fruits that have fallen down from the tree, when they start to break down and decompose, it becomes moldy, but also yeast will start to grow. Yeah, so and that's also how they kind of spread. Yeah. Would you generally recommend generally water is a good medium for the specimen slides, or would you recommend another type of liquid for added contrast or better image quality? So the question is the following: when you make slides, right? I've always added water, and the question is sometimes would it be better to add maybe another liquid, oil, for example? And I'm saying that well, actually the objectives and the microscope has been designed to be used with water and a cover glass, but the question is really not so much the contrast or image quality, but it's rather like this, that some specimens do not mix with water very well. So for example, extreme case bird feathers. If you want to look at a bird feather under the microscope, bird feathers are greasy and fatty and oily, and they will not be covered with water very easily. So if you want to observe small parts of bird feathers or anything else that's kind of greasy, then you probably want to use oil. Not because it increases the contrast or anything, but simply otherwise the bird feathers are not going to spread properly, but they're going to stick together because they want to avoid the water. Yeah, and you don't see it quite as nicely. Yeah. So what I would say over here is what I recommend is not generally water or oil for contrast or but for image quality, but simply because some liquids are not quite as compatible with the specimen. Yeah. So thanks for showing the size difference. Okay, I count yeast every day at work using a hemocytometer, and sometimes I see something smaller than the yeast. So I'm lactic in ethanol. So you're using yeast for ethanol production, okay? So why not lift the cover glass? Yes, I've done that, okay? Microbiologically seen, what is slime mold classified as actually myxomycetes? How do you pronounce it? Slime mold are myxomycetes, and they are eukaryotes. What shall I say? They're not really protozoans in that sense. Or do they? That's actually a good one. Do slime molds officially protozoans? That would be an interesting one. Yeah, but they actually belong to the myxomycetes and basically are single amoeboid cells. Actually, they are protozoans then that are able to aggregate together, and they're able to form this large mass of cells, multinucleated cells, and they're able to crawl around like that. Yeah, but they are eukaryotes. Yeah, so because they have nuclei. Yeah. So what I'm going to do now is I'm going to quickly go through the comments section again. Okay. Let me quickly, I'm just skipping around and looking for those comments that are specifically for me. There's lots of comments. I see my name over here. Paul is responding to Beatrice, not really complex, but the filter must be adapted to your microscope. I think that Micro made a video about this. So I don't know what this refers to. It refers to some of the, maybe polarization or my setup that I have. Yeah. Again, thank you very much. Here at the top here. If your microscope has a filter holder, then you only need a dark field filter. Yeah. I would like to, here's one. I would like to see more dark field microscopy if it's possible. I don't have a dark field microscope, only bright field, but I'm interested in dark field. Thank you for the recommendation. I will show you something now. Okay, just to show you a little bit. So this is a request. So first of all, if your microscope has a condenser, then dark field is quite easily possible. I'm going to show you now quickly what I am able to do, because I do have something like dark field, but only for the low power magnification. I'll show you why. Okay, so this is a bright field. So I've removed the DIC, just regular bright field. And you already know over here, this here now is, to open it, this here is phase contrast. Okay, with a stain cell. Nothing unusual, but in phase contrast, you have a so-called a phase annulus, which is very similar to a dark field filter. Okay, so and look what happens when I leave this phase contrast setting, but I'm going to the top and I'm switching over now to the bright field 40 times, then I get dark field. Okay, so that's basically dark field. So I'm kind of abusing my phase contrast annulus, the phase contrast filter, as a dark field filter. Okay, and it only works in my case for the low power. With the 10 times, you see, I'm back at bright field, and because the size is not correct. Yeah, so I have basically, in other words, I do have just for the 4 times I have a dark field. Yeah, so just saying. And I've made a whole bunch of videos in my other channel, primarily. I've got a couple of channels, actually, microscopy channels. But in my other channel, I actually made some videos where I show you how I've made those dark field filters. And it's certainly something that you should try to make yourself. It can be done quite easily, but you have to be quite precise with that. So I've been actually 3D printing some dark field filters. Simply because it's easier this way, but most people are not 3D printing it, but they're carefully cutting it out, and and then basically you get a result like this. Yeah. So I just wanted to show you, but thinking about this, it actually also looks kind of nice. Yeah. By the way, just because we're talking about dark field, a little bit of interesting thing here. Dark field is very, very sensitive to dust and dirt and contamination. So you see all of those bright dots in the background, right? Look what happens when I switch over to regular. Yeah, you don't quite see them as well in regular bright field. Yeah, but in dark field, because of the contrast, you're seeing dust and dirt much better. And I read somewhere, that's the interesting part. I read somewhere that with dark field, if properly set up, you're actually able to see viruses. Careful, they are below the resolution limit. So you are actually able to see viruses as a bright dot, but the bright dot is significantly larger than the viruses because of the diffraction pattern. So, because the contrast is so large, basically you're actually able to see structures below the resolution limit of the microscope, but the actual structure will appear fuzzy and larger. Okay, so you're not breaking the law of physics this way. Just wanted to inform you on this, that dark field actually is quite powerful in that way, but has the disadvantage that essentially like this, that you're seeing a lot of contaminants, and it kind of makes the image appear a little bit dirty in that sense, because all of the dust and dirt becomes so visible. So I was kind of going a little bit off into technical detail here, right? So I'm going to check again, but otherwise I would say suggestion protists under adverse oxygen conditions, Daphnia reproduction. Thank you for the suggestion. Maybe there's a possibility to kind of control the oxygen concentration somehow of the protists. Yeah. Okay, people. I think I'm going to call it a day. Call it quits today. Yeah, I hope it was interesting again. I personally like doing these things here. Right? I hope, quite sure that from the comments that I'm seeing here, that you're also quite getting involved. Again, I cannot thank you enough. Thank you very much for the donations. It's really nice of you. And Yeah, maybe I'm gonna do another thing, another advertisement. I do have this influencer shop on Amazon. Yeah, so where essentially I'm getting a commission every time something is sold. I have to keep on. Have to keep the products updated a little bit because I've not been doing that recently. But that's also one of the way how you can contribute to the channel. And what I'm doing is I'm using those donations again to buy new microscopy equipment, or for example those petri dishes I ordered, yeah, and things like this. So I'm trying to feed it back a little bit into the video, into the channels here. I'm going to call it a day today. I wish you all the best. Okay, hope to see you again next week. I'm going to be thinking of another topic again. Happy microbe hunting as always, and bye bye.